Welcome to the biodiversity seminar of today. I'm very proud to host this seminar and introduce Debbie Leigh to you. Actually, I think Debbie does not need too much of an introduction because she has been here almost 5 years already at WSL. But I tried to give you the most important milestones of her CV so far. So Debbie has done her studies in London and Edinburgh, and then came to the University of Zurich to do a PhD in Lucas Keller's group on the conservation genomics of alpine ibex populations. After that PhD, she did 3 different postdocs, one in Canada at the Queen's University about seabird genomics. And then she came to WSL in 2019 to work with Danny Riegling and on virus genomics, virus population genomics. And in 2021, I think she moved to our group to work within the biodiversity project ACORN, with me. And in this project, Debbie was responsible mainly, among others, for the common garden experiment, especially the one we had here in Birmenstorf. So planning, execution, a lot of data collection, sequencing all the individuals. And finally, she will show that today, connecting the traits with genomic data. But Debbie has not only been very active in pure scientific activities, she's also quite active in outreach and institutional outreach, I would call that. And she's, for example, member of the Powell Center Macrogenetics Group. She is a steering committee member of the IUCN Conservation Genetics Specialist Group, and she's also a member of the GeoBon Genetic Composition Working Group. So not only active in pure science, but also in institutional outreach a lot. So the floor is yours. We are very curious to hear.
Thank you. Thank you very much all for coming, and thank you, Christian, for that really kind introduction. So I'm really excited to be here today because it's quite rare that one has the opportunity to talk to so many of your collaborators, both in the room and online. This project, as Christian mentioned, was done on site and has really benefited from a huge amount of the expertise that we have at WSL, everything from discussions about methods to lending us equipment to, just stopping to say hi when we were in the field. So I'm really happy to have the chance to show people what we've done with our work and where we're taking the project as we move forward. From a personal perspective, Christian also mentioned that I've been here for 5 years, and actually my time at WSL is ending. I'm a temporary scientist, so in the next few weeks I will be leaving, and I will be, negotiations going well, undertaking an associate professorship in Germany. So it's really nice to have the chance to bookend my time here by showing you some of my work. So how can we prepare forests for the future by characterizing white oak climatic adaptation? Across all of biodiversity, species are encountering very different environments from historical norms due to human-mediated land use change and climate change. Species are basically existing in environments or habitats that are completely different from their historical norms. Many species are now quite ill-suited to these new environments, and so they're suffering fitness consequences, fitness loss in particular from these changes. And in order to avoid decline and ultimately extinction, species have a couple of options available to them. The first is that they can migrate with their historical climatic niche as it moves upwards in altitude or upwards or downwards in latitude. The second option they have available to them is to change in response to the changes around them, so to adapt in response to these new changes. If they fail to do either, they of course run the risk of fitness loss and ultimately extinction. But even if they succeed to do either of these things, the changes are so big and so rapid that many species may still go extinct because they just can't change or adapt fast enough. And forest trees are not immune to these changes. Here you see a map of European forest cover. And I just wanted to start by saying that forest trees, particularly common forest trees in Europe, are quite abundant. So we're not talking necessarily about the extinction of specific species of trees within Europe, but we are talking about the extirpation, so the local extinction of some populations, and quite serious harmful effects of climate change to forests. One of the most pertinent changes that's ongoing and will only continue to worsen in the next century is the increase in frequency and intensity of drought. Droughts, as we can all observe, are just becoming more and more common. As they become more common, more intense, we start to see increase in canopy mortality. So tree leaves and canopies are just dying back due to the water and heat stress, as shown here. With increased intensity and frequency of drought, forest canopy dieback is becoming more and more common. Now, coupled with canopy dieback in quite extreme situations is total tree mortality. And hotspots for this for the next century are shown here. Oh, the pointer is a little bit on its own tiki tour at the moment. Here we go. In red, which is pretty much all of Southern Europe. And what this will do is basically jeopardize the ecosystem services that many forest trees provide. They will maybe provide lower quality lumber, less cooling effects. Of course, their ability to act as keystone species is also compromised. Now, we often talk about the effects of climate change in the future tense, but unfortunately, the effects of drought on trees are something we will have all had the opportunity to observe within our own eyes. And by drought, I mean climate change mediated drought. So 2 years ago in the summer of 2022, so this is but one summer ago, we had a very extreme drought here in Europe that I'm sure we all remember. And this was severe enough that it started to affect tree species other than beech, which is typically very water sensitive. Here you see a Quercus robur individual. Sorry, the quality in the room might not be great, but you can see that it looks like it's in autumn foliage. We have a lot of dieback. But this picture was taken on the 1st of August. So this is drought-mediated cell death or tissue death. And you'd think, oh, maybe this picture, it's in like the north of Spain, south of Italy, somewhere where the drought was really intense. Brittany, in my in-laws' garden, so the north of France, a very wet, cool historical region. So this is showing that climate change mediated drought is starting to impact species really in the heart of their distributions. So what can we do to help forests in the future? One tool that we have in our toolbox is assisted migration. So facilitating and speeding up these natural migration and adaptation processes that species can do to help maintain fitness. Now, there are many different flavors of assisted migration, depending on who you talk to. And the one we're going to be talking about today is the movement of reproductive material like seeds within a species' existing range. So why would you move individuals within a species range? Well, first, to facilitate long-distance migration, moving populations quite further than they could naturally. And the second, the idea is basically to facilitate adaptive transfer. So to move populations that might already be adapted to future climatic conditions, to help establish trees that are adapted to future climate scenarios, and hopefully have them interbreed with the local population so these adaptations can resonate and spread. Now, we're quite a diverse group of people in the room, and I assume so also online. So I just want to walk through this with you to really make sure this kind of approach and the impacts are clear. Typically, with assisted migration, we talk about a south to north transfer. So in the idea of oaks in Switzerland, we might be thinking about maybe moving individuals from Greece or Italy to Switzerland, because the idea is that in Greece or Italy, they've historically had much drier climates, much more experience with drought, much more experience with heat stress. So you're assuming that these populations have adapted to these types of conditions. And by moving them up, so moving populations rather than changing species, you're transferring these adaptations up, establishing these pre-adapted populations and hopefully allowing their adaptations to spread into the natural stance. However, recent evidence from oak trees in North America and a few other species around the globe has suggested that some species of trees can show very, very specific population local adaptation. What that means is that each population or genetic unit might use its own gene networks or different genes to respond to the same climatic stressor. So here we have a expression analysis done on valley oaks. Each line here is a population, and the different rows are different gene network responses, and the colors show up regulation and down regulation. And what it shows is basically in response to the same stressor, this was water stress, we see completely different changes in regulation depending on the population that you focus on. So very, very highly localized responses to quite an important climatic stressor. So this, of course, has implications for assisted migration, because if you're moving individuals that use completely different genes or gene networks to respond to the same stressor, when they start to interbreed, they might produce offspring that are quite maladapted, that just have a breakdown of these adaptive complexes. So one solution that has recently been proposed is to do kind of micro-assisted migration, to move individuals from, say, historically dry soils to historically wet soils that are going to start to become drier with the increase in frequency of drought. The idea being that on the dry soils, individuals might have developed highly localized adaptations to drought, but the close proximity will mean that they have very similar adaptive networks for the other genes, to the other stressors that they experience. So this would allow you to do assisted migration and maintain forest health without potentially disrupting these adaptive gene networks. But the crux of this method really depends on adaptation being present at such a fine scale. And we have to remember that in Europe, forest trees are quite common, they're quite abundant, they're also moved around a lot by people, and they have a lot of wind-mediated gene flow. So they may not show such fine-scale adaptation. Answering these questions is really the crux of the project that Christian proposed in the Biodiversity Project, this ACORN project, which is our running title. We're trying to understand the scale and structure of adaptation specifically to drought in European white oaks, focusing on 3 species, Quercus robur, Quercus petrea, and Quercus pubescens, which are very culturally important. I'm sure we all know oak leaves you can see everywhere, carved into wood and things like that. They also have huge importance for lumber industries, and of course, they're very, very important keystone species for forest ecosystems. The ACORN project is taking 2 approaches. The first that I'm going to talk about today is this GWAS Common Garden approach, where, well, I'll basically go on to what we're doing in the next few slides. The second is something I'm not going to talk about today, but maybe someone could invite someone from Boku to give a BD seminar, or maybe Christian can talk about it next year when we finish this analysis. But it's basically a classical scan for selection across the landscape, so a GEA analysis. So what am I going to talk about today? Well, what we've been doing to try and understand the scale and structure of drought adaptation in Europe is basically to run common gardens. Common gardens are essentially standardized provenance trials. You take individuals of a known origin and you grow them in the same environment, shown here by the green square. So all of these individuals might have different genetic backgrounds, but the environment is the same. And this allows you to disentangle the impact of genes on specific traits. So you can see that any differences you observe in the common environment are likely due to genetic impacts. And if you consistently see these genetic impacts in individuals from certain provenances, you can make inferences about whether or not the trait is adaptive. In the ACORN project, we've established 3 common gardens. The first was in the Turkish botanical gardens in Ankara in Turkey. The second was here at VSL in Zurich, Switzerland. And the third was at Boku in Vienna, Austria. And they look like this. So you can see very different environments. The idea behind the provenance selection was really to test this local versus regional question. So what we did was to take provenances that were very close together, about 10 kilometers apart, with known differences in soil moisture conditions, and grow them in the same environment to disentangle this local hypothesis. But we were also still interested in these regional scale adaptations, so we did the same thing, but for individuals from southern provenances as well. Now, I say we did that, but oak trees are masting species. So our ability to do this was really constrained on seed availability and mast year. So we were only able to get this kind of optimized sampling design for Quercus pubescens, shown here. For Quercus robur, we were able to get this regional approach in northern provenances, but southern provenances were much more sporadic and spontaneous, and were basically acorns kindly donated by collaborators. We followed these trees for 2 years, starting in 2021. As Christian mentioned, we planted them. In 2022 was our first trait year, and 2023, last year, was the last year of taking traits for the experiment. This is what they looked like in 2023. We measured a litany of different traits, basically everything we could, with perhaps the most important for drought being delta 13C here. And I want to acknowledge that we definitely measured traits that were not necessarily linked to drought responses or climate change responses, but we thought we'd just make the most of the gardens and measure everything that we could because we had them on site. Coupled with the trait analysis, as Christian mentioned, I am also a genomicist and a bioinformatician. So what we did was to genotype all of the individuals across all 3 common gardens. So this is about 1,200 individuals, and we genotyped them using whole genome sequencing. To make that affordable, we used low coverage whole genome sequencing. And what we did with this was basically to do a genome-wide association study. What we were interested in doing was linking genotypes, so the presence or absence of mutations at specific parts of the genome, with phenotypes, so the presence or absence, in this case, of a disease, in our case, of maybe specific drought responses. The idea being that by linking the two, you can potentially find the genes that control the trait of interest that allows you to maybe scale up the study or understand through genotypes whether or not populations might be drought adapted. Now, just briefly, I mentioned we use low coverage whole genome sequencing, and I just want to touch upon what that means. When you interpret the results that you see, these are not hard call genotypes. So we don't know for certain if there is a mutation at any particular location. What we're working with is the probability of mutations at these locations. And it's just important to interpret some graphs. So moving on to the results, the first thing that I wanted to show you is what we could figure out just from basic population structure. So looking at genetic similarity across the provenances. So here we have Quercus pubescens. And what we can see is that already between Turkey and Central Europe, we have very strong population structure. The closer the individuals are together, the more similar they are. So the further apart, the more different they are. And we can see that Central Europe has one kind of cluster here that separates on axis 2, and Southern Europe has one cluster here, but the biggest difference is between the two. And this marries with trait differences that are very, very visible to the eye within the common garden. So as you can see from the pictures here, the two populations or two genetic clusters show quite profound differences in leaf genotype. So, or leaf trait, sorry. We see individuals in the south of Europe are very curly. They're actually almost velvety in hair-like. And the individuals in the north of Europe are very flat and much more waxy or sheen-like, and the hairs are only visible under the eye loop. Within Quercus robur, we also find quite strong population structure, with actually the strongest being here between southern Italy and the rest of Europe. And this is quite distinct. It's collapsed a bit on the axis, but there is a very, very distinct break here. And this again marries with trait differences within the experiment. So individuals from this provenance show basically quite completely different phenology from the rest of the populations. They're essentially functionally evergreen. And this has been contributed by a recent study on this population by our the collaborators who lent us the seeds to be due to the fact that the individuals from this provenance, which are shown here at the very bottom of Italy in Bosco Pantano, these individuals are basically representative of a distinct glacial relic population. So this population is made up of about 70 individuals that have been separated from the rest of the European populations for up to 200,000 years. So this type of information is really, really useful already for conservation managers, practitioners, foresters, for example, because you might think, okay, yeah, let's take from the south of Italy and move it upwards. But if these have been separated for so long, this could have a number of impacts that we don't really think about. So it really already gives us quite a lot of valuable management information. So what about traits and what about the GWAS? Well, as I mentioned at the beginning, one of the most important traits for drought is the delta 13C stable isotope. What is the delta 13C stable isotope? So I see some blank faces, so I will explain this. On the underside of a plant's leaf are stomata. These are pores that are the site of gas exchange for photosynthesis. So basically where the plant gets its CO2. These stomata are also sites of significant water loss for the plant. So when plants start to experience drought, they close their stomata. Now, normally plants are really picky for which carbon isotope they use for photosynthesis. They preferentially use the carbon-12 isotope for photosynthesis in the atmosphere. But when your stomata are closed, you have to exhaust your supply of carbon. So they start to use the carbon-13 isotope. This leads to a lasting signal in terms of the ratio of 12 to 13C that's incorporated into the tissue in the plants. And if you look at this ratio, you can infer how much the stomata have opened and closed over the period that you're looking at, which gives you insight into drought responses. So we decided to look at the delta 13C ratios of a single leaf. And I wanted to show you this to explain why it was just one leaf. So you can see in the first year, our trees were pretty tiny. So taking more than one leaf would have been a bit disastrous for some of them. And we took it in September, at the very beginning of September, to try and capture the entirety of the growing season. So let's look at the 13C results for Robur for 2022, which is the first year of life for these trees. What we can see is that the delta 13C values show a strongest association with precipitation seasonality. So these local differences in soil moisture didn't significantly affect delta 13C values. The higher the values, the more populations opened or individuals, closed their stomata. So these individuals here would have closed their stomata the most because the values are the highest, which makes sense because they were the individuals grown in Turkey. TR is the Turkish botanical garden, CH the Swiss, and AUS, Austria, the Austrian one. So we can already see the difference in environment here. Now, when we run it through a quick mixed effects model or an animal model using a pedigree, we can see that the most significant factor affecting delta 13C in Robur seems to be the seasonality of precipitation. So the regularity of precipitation that the provenance of origin has had. But the patterns really go against our expectations, with populations with higher seasonality, so less regular precipitation, showing lower delta 13C values. So they seem to have their stomata open more. What does this mean? Well, it's not really clear, but it suggests that either they have more responsive stomata, smaller stomata, or they might have a trade-off where they're maximizing their water use when water is available. As I mentioned, we can also use the low coverage whole genome sequencing to start to understand the genes that are involved in this response using a GWAS. Now, typically with a GWAS, I'm just showing you this again to show you what this type of plot will look like. By finding the peaks, so these parts here, you can find the genes of interest. Now, this is a very idealized GWAS for humans and a binary trait. So none of our GWAS look like this. They look like this. But this is already really exciting. So you can see the different lines here. GWAS, there's many, many different discussions that I would be happy to have with many of you for hours over significance thresholds and how you deal with multiple testing. Some people use a 5 to the 8 threshold. Some people use a 5 to the 1. Some people use Bonferroni corrections. Some people use Benjamini-Hochberg, so on and so forth. The results that I'm going to show to you today are basically all significant and at above a 5 to the -7 threshold. So already we can see this really great peak. Actually, it's marginally non-significant at E times 5.4 to the -7, but it's close enough that I wanted to discuss it with you guys. This is a very interesting peak and quite convincing because we see several SNPs that are very close together in one peak, showing an association with the delta 13C values in Quercus robur. And actually, this is an auxin efflux carrier gene that is involved in the elongation of roots and root hydrotropism, which basically means it's how the roots, shown here, turn towards water. So this is a very interesting candidate for drought response. We can also see another significant hit here. Basically, we're looking at the top of the range here, which is a ligase involved gene, potentially. But there aren't very good hits here because, of course, we're not working with Arabidopsis. So we can't always be sure what SNP does what within the genome. If we look at this particularly highly significant SNP and we look at the differences in trait value, so here you see the delta 13C and here you see the different genotypes, we can see there is quite a significant difference or a significant decline across here. It's not statistically significant, sorry, I should say substantial decline. And you can see there are fewer individuals with this TT genotype. This is a product of the low coverage whole genome sequencing. You will always have a bias towards the reference genome. If we look at Quercus pubescens, rather excitingly, we see a completely different scale of adaptation. We see that by and far, the most significant impact on the delta 13C values is the expert classed local soil conditions, with individuals taken from provenances that have these historically dry soils showing much higher on average delta 13C values. So this suggests that there is quite localized adaptation in Quercus pubescens. And when we look at the GWAS, we see some significant hits. I think this one here is one of the most significant. But what I wanted to show is that basically we have a number of different hits that potentially signify many different genes being involved in this response across the different populations. So overall, using this approach, how can we translate this into something that's meaningful for foresters? Well, firstly, these results are really preliminary, so please don't hold me at my word. But what we can show is that potentially Quercus pubescens shows much more local adaptation to drought than Quercus robur. Why Quercus robur shows this opposite pattern is not really clear to us. It's something that would need to be explored further. But ultimately, this has quite interesting implications for the management and movement of individuals. So I still have a little bit of time left, so I thought I would show you a couple more traits and what we found there. So another trait that we measured with our single leaf on the advice of Charlotte Gossard is specific leaf area. So it's the dry mass relative to the area of the leaf as measured by something like image J. And here, really excitingly, we have a very, very significant hit that is in a cell wall biosynthesis gene, potentially suggesting that this gene is somehow related to differences that we see across SLA within key different individuals. We also, throughout our experiment, kind of came up with our own traits. So while weeding the garden and measuring the height and things like that over the first year, Christian and I noticed that many of the trees showed what we called this bushy phenotype. They just basically looked like a bush. And we were really interested in this because many of them seemed to be from the same provenance. And it was quite striking. So we kind of came up with this bushiness index, which is the number of leaves divided by the total height of the tree. And I just need to shout out our interns from this summer for this trait because they counted all of the leaves on our trees. And some of the trees at the end of this year had over 1,000 leaves on them. So they did a great job, but all was for good because we found, again, some really interesting hits. And actually, this one is already in an identified QTL for branching architecture in oaks. So this is really nicely validated by another study that found the same genomic region of interest. And we see quite a big difference in height that's consistent across, in bushiness, sorry, that's consistent across the three gardens. And already you can see that the three gardens also show very big differences in total height as well, which is something that we're going to explore. So we're reaching the end now of the talk, and we do have some next steps. And a lot of this analysis is still ongoing. So if you have any feedback or want to discuss it with us, please come find us. I'm here hopefully until the end of April. One thing that is going to be ongoing for a little bit longer than April is Simone's master's thesis looking at chlorophyll and non-chlorophyll related pigments and phenology and doing a GWAS and an assessment of this across the common gardens. We're also hoping to do a more sophisticated analysis of traits. What I've shown you is a combination of linear mixed effect models and MCGLLM that basically incorporate relatedness using pedigrees. I'd like to start to incorporate relatedness using genomic estimated relatedness, because some of the acorns we have are definitely not the offspring of the mothers we thought they were, because forests are not tidy, you know. And one of the most important things that we're going to do is basically create outreach and practical tools to explain how to use these results in terms of assisted migration or drought scanning, which will come when we have both parts of the project completed and can synergize them together. So unfortunately, you can't go visit the garden anymore. I had hoped you might be able to at the end of this talk, but the garden, as you will see, has been dug out and the trees are sadly in their little plastic bin bags. But fear not, they're not going in the trash. They're being moved to Favre in Freiburg. So with that, I bring a close to my project on Quercus here at WSL. As Christian mentioned, I've worked on many different things in my time here at WSL that all focus on synthesizing and understanding genetic diversity at different scales. And I'd be really happy to talk to you about those at another time, but I think half an hour of me talking is enough for today. So with that, I just need to take a moment to thank the many, many collaborators who've made this work possible, some of whom are shown here, some of whom I don't have pictures of yet. And I would be more than happy to take your feedback and have any questions from you. Thank you very much for listening.
Thank you. Thank you so much, Debbie, also for being so much on time. So we have a lot of time for questions. I will not ask questions because I'm too much involved in the project. So please ask your questions.
Hey, thanks for the talk. So what made you decide on not measuring like root-related traits with, for example, a subset of the trees every year? Because especially with the GWAS hit, it would be super interesting to have some root traits measured, right?
I mean, the issue is that because they're planted in the ground rather than in pots, it would be too destructive. So we had thought about maybe taking root samples for stable isotope analysis when we dug them out now, but we just don't have the capacity. So it was just practical limitations, basically. Yeah.
Any other questions?
Thanks for this talk. It's really excellent. I was wondering about the climate at the Three Common Garden during the experiment. I mean, I assume that in Turkey it was generally hot and dry, but how were like Austria and Switzerland different? And like, would that have somehow influenced the outcome of your results?
Yeah, for sure. So Turkey was very hot and dry. We did water in the first year and then a little bit less this year because they're seedlings. So if we hadn't watered them, they would have died. And we did have very low survival of the northern provinces already in Turkey because of that. Yeah, it was by far the most extreme. And I think I have a picture if I can get to it. It's also, the site is pretty extreme. It's in the middle of Ankara. So it's like, yeah, I don't think I'm going to find this picture. Never mind. It's very warm. The one in Baku, as you can see here, is much more shaded and forested relative to ours. So we think that might have also contributed to the lower growth rate because they're much more in the shade. And we had a much higher incidence in the first year of powdery mildew fungus on the leaves. This is an invasive fungal pathogen from mango skins. And we treat the trees for it because it's not a natural part of kind of the experiment, and it's not present in Turkey as well. But it seems like the treatment wasn't 100% there. So that might have also impacted growth. So yeah, the climates do differ across the two sites quite a bit. Yeah.
You now showed some results on Quercus robur and pubescens, and I would have a rough guess that Petrea is somewhere in between. Can you elaborate a bit on that? Or?
Yeah, so what I forgot to mention is that Petrea, basically we were not able to get any pure Petrea southern provenances, and the provenance that we did get had very low germination success. So we only have northern provenances for Petrea, which is why we've excluded it from the bulk of the analysis that I'm presenting. Yeah, it's just, we don't have the numbers there to make any concrete conclusions. So we chose to focus on these two species instead.
Okay. Thank you for the presentation. A very naive question. I'm wondering how you select the traits you want to associate with the GWAS.
Yeah.
Because, yeah, sorry.
No, go for it.
Because if you, do you test for significant difference, for example, between the population and then you go for the GWAS? Because otherwise, if you have no difference in your population, in the traits, I mean, does the GWAS mean something after?
Yeah, it does, because it's still, the GWAS doesn't relate to the adaptive capacity, right? The GWAS just links phenotype with genotype. It doesn't give you any information about whether or not the trait is adaptive. That's, you need the linear models for that. We basically chose the traits that we measured that made sense. It's very easy for us to run the GWAS, so we just threw everything in. And then what's interesting is maybe what you don't see or what you do see consistently across the different models. So for specific leaf area, for example, for the result that I showed you, we have this very high hit that's highly significant. But actually in the linear model, using precipitation and temperature metrics for this trait, we find no significant factor that affects the trait. So I think you have to dissociate in your mind the trait analysis looking for adaptive variation in the traits with the GWAS, because the GWAS is basically saying this difference in trait is potentially driven by this gene. And then maybe looking at the frequency of the gene across space would give you insight into whether or not it's adaptive.
Thank you very much for the talk. It's really enjoyable to listen to you.
Oh, thank you.
My question relates to this assisted migration. I wonder, is this something that foresters practice already, or they support the idea? Or is this something only discussed in science as a theory that this would be useful in future?
It depends on the flavor of assisted migration. So one thing that is very widely practiced by foresters and landscape architects as well is movement of species outside of their range. So like, for example, planting Quercus cerris, which is a southern Quercus species up in the north of Switzerland now for drought responses. So this is a type of assisted migration where you're forcing the range upwards. Movement within a species range is practiced, I think, in North America. My knowledge of the practice here in Europe is that it's not frequent. Do you agree with that? Yeah, it's infrequent, but by maybe creating fact sheets that explain it and explain how to do it in such an iconic species, we hope that we could potentially show people it's an option that is secure. So, yeah.
Thank you.
Eva's very we have time, guys, don't worry. Yeah. Let's say that we start doing my question is, or it could be and are people for I imagine it would be movement of seedlings because of the way oak work is done. It tends to be that you collect seeds from specific seed orchards and then you grow the seeds up from the specific seed orchards and then you plant them. Movement of seeds would be like quite high loss because you'd get a lot of the seeds that would be eaten by mice, right? So. In terms of movement of associated pathogens, that's something we definitely had to consider. But I imagine, yeah, you would probably need the plant passport just like you need it for anything, right? So I guess so. But cross country, like setting up new seed sources or things like that, that seems like quite a big aim. I'm not sure how practically realistic that will be in the next few years. It depends on the convincingness of our results, I suppose. Yeah. Yeah. Mm. Yeah. Yeah. Sorry. I can repeat it. It's okay. So Eva's worried about there being a gap in science or a disconnect where people who work on assisted migration don't worry about pests, and that's probably true. I mean, I think if with the local movement of individuals, that's clearly not going to be a problem. But yes, if you do need to do large scale movement of individuals, that might be a problem. Yeah.
I'm not a population geneticist, so I ask a very naive question. So I understand individuals or a population on the warm, dry edges well adapted to these conditions and is tolerating drier conditions, sites than one from the wet, colder edge. Wouldn't it then be sufficient to just say, we need to find out where do we have in the future the comparable climate? So what does genetic add? So why do we need genetics instead of just trying to find out what are the climate transfers that we need to do?
So you don't, just because an individual is present in a certain climate doesn't mean it's adapted to it. That's particularly true in the Valley Oaks, for example. They've already shown that the populations are already maladapted to the temperatures that they experience. And and so that's something to already consider. Like species may not have had the time even to adapt to their historical climates, especially species with such long generation times. I don't think you would be off in taking this approach, but what genetics adds is if we just look at population genetics, for example, so this type of analysis where we looked at the clustering, you can already say, okay, yes, movement from large scale approaches is possible, but look, these are very, very different. So if we move these individuals from the south to the north, once they start to interbreed with the local population and the individuals that still survive, because assuming some individuals will survive the future climatic conditions that are present, the progeny might have very, very reduced fitness. And this could create a population that's basically unable to self-sustain because of the reduction in fitness. So that's one thing we can do. With basically understanding the genes that are involved, this is more than just like an Arabidopsis exercise or things like that where we're doing crop improvement. There's definitely ways that you could use that to scan using broader like SNP chips and scanning populations, say, do we think this is adapted to drought based on previous evidence? Maybe it's a good seed source for this, things like that. I do think there's a lot it can add, but I agree, I see the concern.
Just a follow-up, wouldn't you need to be careful to not interpret adaptation too linearly to one or few factors that you look at? Because usually adaptation is in many different directions.
Exactly, yeah.
And it may not always be to the favor of performance for exactly this characteristic that you look at.
Yeah, totally. So that's also why we've added the phenology and the like the chlorophyll and non-chlorophyll related pigment traits that Simone is doing, because these are, of course, not necessarily drought related, although chlorophyll can be. this is really to start to see if other traits could show significant adaptive variation that has to be considered. So for example, would we have to altitude match, things like that? Yeah.
Maybe I can just add that there are now several studies, probably there's a publication bias, I don't know, but there are several studies showing that if you take into account genetics and climate into your transfer distance analysis, you have an improved fitness when only taking climate into account.
Just quickly, how do you think your results would change if you were to follow these seedlings beyond 2 years?
Yeah, it would be really nice. So unfortunately, we ran out of money, basically. So already I looked at a subset of individuals that have stable isotope data for this year versus last year, and it was two completely different, 2023 and 2022. And even though we watered in 2022, it was so hot. I mean, we had to put straw down to reduce the surface air temperature because the soil was cracking. So already we see quite low correlation across the 2 years. So we might start to pick out different genes and things like that. And yeah, I'm sure there would be a number of traits that would start to change, especially related to phenology. Like Jan is quite convinced that phenology in oak seedlings is very shallow, and the effects only start to build up as they get older. So that would be something that could also potentially change throughout time. Yeah.
I think like the difference in the populations that you showed in this PCA would somehow.
Oh no, the PCA is consistent because that's fixed, right? That's the genotype of the individual. So those differences will remain because that's genetic. That's based on genetics. It would be the traits that would change. Yeah. Yeah. Anything online?
Hi, Debbie. So thank you for your talk. It was very understanding. I just have a question regarding the saplings. So you said that you had to water them all the time because of the weather and because there wasn't raining a lot. So how do you think would they survive in the wild if you plant them somewhere?
Yeah.
Because I mean, it's not really. I think it's kind of falsifying the data if you're not. If you had to put a lot of effort to make them even grow. So how do you think will that?
So we didn't put a lot of effort in. Like the most we watered them was like once a week, maybe twice a week when it was like 35 degrees. So we did minimize water as much as possible, but it will definitely be an impact and we have to kind of interpret our results based on that. I think that you have to also remember that in nature, it's quite rare that an oak seedling is planted into bare earth in an open field, because typically they would be in forests, like under leaf mold that keeps them nice and moist and creates like this little microclimate for them. So I think that even if we hadn't watered them, it would have been an unnatural environment. So it's a bit of a trade-off that unfortunately was one where we had to lean on the side of watering. Otherwise, my sample size for this year would have been like 2. So yeah. Thank you.
And it's amazing that you can perform controlled experiments with your study species. It's just so great.
Yes, it's the first time I'm able to do this.
It's amazing. So, which made me thinking, would it make sense to perform some gene expression analysis to check for differential expressed genes?
Absolutely. Yeah.
Have you?
Yeah.
Yeah. I
Totally.
know. I know.
Yeah.
have you get samples just in case, or
no?
Okay.
No, we didn't think about that. Like I have to say, sampling was not trivial. So the sampling for DNA, you have to do it really early in the year, and we had to do it 2 years ago when it was like 35 degrees, and so we were like it takes like a week basically to sample any trait in the experiment, maybe 2. So we just didn't have the manpower to take just in case. I had really wanted to do that and take like root samples, but yeah.
We have one question here, like a more general one. Somebody asking, thank you for the information. I'm part of a group that volunteers for different environmental projects. We have yet to find a group that is planting trees. So 2 questions. Would there be a need at VSL for future help in planting trees or seedlings for studies? And are you aware of any organizations that might be planting trees in the canton of Zurich?
We always need help. And I think the other common gardens definitely also need help. I think we have quite a few common gardens that VSL is part of, but I don't know if they're in the canton of Zurich. In terms of organizations in the canton of Zurich that are planting trees, do you know any? For Quirk? Yeah, maybe Grünstadt Zurich. Otherwise, I knew when we were checking for foresters to speak to, the foresters in, I think it's in Biel, are doing a lot of Quercus pubescens planting to try and increase the frequency to drought-proof the forests there. So there are, and in Canton Geneva as well, I think they're doing that. So it's very localized. Yeah. Cool. Okay, well, thank you all for your excellent questions. That was really fun. So thank you for coming.
Thank you, Debbie. So if you want to continue discussing, come